Alright everyone, welcome back. So now that we understand that DNA is in fact our transforming material, it came time to try and figure out what did the structure of this DNA material look like. And if we remember that DNA stands for deoxyribonucleic acid, we know that it's now the sequence of the DNA nucleotides, how those letters get put together in the nucleus that ends up giving us that genetic code. But it took until the mid-1950s for us to actually understand what the DNA molecule looked like. And that structure was discovered by two scientists. They were really model makers at the time, Watson and Crick. And they figured out the structure of the double helix, the DNA, based off of this woman, Rosalind Franklin's picture called Photo 51. All right. So these two gentlemen were working in the 50s trying to come up with a structure for DNA, and they were putting all sorts of pieces together. They understood the basic building block of a nucleotide, but they didn't know how they fit together until Rosalind Franklin's picture. And so once we get to the 1950s, these gentlemen are given lots and lots of credit for figuring out that structure.
The discovery of the structure of DNA was one of the most important scientific achievements in the last century, in human history, in fact. The now famous double helix is almost synonymous with Watson and Crick, two of the scientists who won the Nobel Prize for figuring it out. But there's another name you may know too, Rosalind Franklin. You may have heard that her data supported Watson and Crick's brilliant idea, or that she was a plain-dressing, belligerent scientist, which is how Watson actually described her in The Double Helix. But thanks to Franklin's biographers, who investigated her life and interviewed many people close to her, we now know that that account is far from true, and her scientific contributions have been vastly underplayed. Let's hear the real story. Rosalind Elsie Franklin was born in London in 1920. She wanted to be a scientist ever since she was a teenager, which wasn't a common or easy career path for girls at that time. But she excelled at science anyway. She won a scholarship to Cambridge to study chemistry, where she earned her PhD, and she later conducted research on the structure of coal that led to better gas masks for the British during World War II. In 1951, she joined King's College to use X-ray techniques to study the structure of DNA, then one of the hottest topics in science. Franklin upgraded the X-ray lab and got to work, shining high-energy X-rays on tiny, wet crystals of DNA. But the academic culture at the time wasn't very friendly to women, and Franklin was isolated from her colleagues. She clashed with Maurice Wilkins, a lab mate who assumed Franklin had been hired as his assistant. But Franklin kept working, and in 1952, she obtained Photo 51, the most famous X-ray image of DNA. Just getting the image took 100 hours. The calculations necessary to analyze it would take a year. Meanwhile, the American biologist James Watson and the British physicist Francis Crick were also working on finding DNA's structure. Without Franklin's knowledge, Wilkins took Photo 51 and showed it to Watson and Crick. Instead of calculating the exact position of every atom, they did a quick analysis of Franklin's data and used that to build a few potential structures. Eventually, they arrived at the right one. DNA is made of two helicoidal strands, one opposite the other, with bases at the center like rungs of a ladder. Watson and Crick published their model in April 1953. Meanwhile, Franklin had finished her calculations, come to the same conclusion, and submitted her own manuscript. The journal published the manuscripts together, but put Franklin's last, making it look like her experiments just confirmed Watson and Crick's breakthrough, instead of inspiring it. But Franklin had already stopped working on DNA and died of cancer in 1958, never knowing that Watson and Crick had seen her photographs. Watson, Crick, and Wilkins won the Nobel Prize in 1962 for their work on DNA. It's often said that Franklin would have been recognized by a Nobel Prize if only they could be awarded posthumously. And in fact, it's possible she could have won twice. Her work on the structure of viruses led to a Nobel for a colleague in 1982. It's time to tell the story of a brave woman who fought sexism in science and whose work revolutionized medicine, biology, and agriculture. It's time to honor Rosalind L.C. Franklin, the unsung mother of the double helix.
And when we look at DNA structure, here's the structure of a nucleotide, right? And your nucleotides are made up of three major components. You've got your phosphate group, which is up here. That's attached to the fifth carbon that's part of a pentose sugar, all right? And since we're talking DNA, that pentose sugar is deoxyribose, all right, which means it's only a hydrogen here. When we get to talking about RNA in a few days, ribose has an OH group there. And then we also have a nitrogenous base, and those bases can either be adenine, thymine, cytosine, or guanine, all right? And this is a general structure of a nucleotide. Now, when we go to the next slide, we're going to see that some nitrogenous bases are a single ring like this. Others are going to be a double ring, okay? Put together by base pairs, okay? So when we look at DNA structure, it sort of looks like a twisted ladder. And your base pairs are what makes up the rungs of the ladder, so where you would step. And those nucleotides are held together by hydrogen bonds. And your base pairing rules come from the fact that guanine and cytosine, all right, have to make 3 hydrogen bonds between them, okay, in order for them to have a stable configuration. Adenine and thymine, however, have to base pair because they can only make 2. So I can't put an adenine with a cytosine because then cytosine can't make this last hydrogen bond in order for it to be stable. Reverse would be true if I tried to put guanine and thymine together. That last hydrogen bond for guanine couldn't get made. Okay? So if you don't already have this memorized, A goes with T, C goes with G. Okay? And it comes down to the fact of how many hydrogen bonds they can make in order to be stable with each other. What's interesting, though, then, is we also ended up discovering that for every pyrimidine, it had to bond with a purine. All right, so if I know that I have 100 bases in a DNA molecule, then I know that if I have 50 purines, I have to have 50 pyrimidines. Okay. The other way this works is that if I know I have 10 adenines in that molecule, it means I have to have 10 thymines. Well, that takes up 20 bases. All right, leaving me 80 left, which means 40 have to be cytosine, 40 have to be guanine. So understanding this base pairing rule allows us to then understand how many of each base we have, so long as I know one of them.
All right, so when we take a look at the actual monomers, we're talking that nucleotide, right? But when we start stacking all of those nucleotides together to actually make a full DNA molecule, we're looking at a double-stranded molecule. So it kind of ends up looking like a twisted ladder. What we talk about is a double helix. So those 2 strands of nucleotides get connected with a sugar phosphate backbone, and then in the center where the rungs of the ladder would be, if you use that analogy, are your complementary nucleotide base pairs. So A always matches with T, C always matches with G. All right. So if I come up and take a look at just kind of a strand here, half a strand, and I take a look, here is a nucleotide. Right? So this would be your nucleotide. And it's really important to pay attention to what's connected to what. So I've got my phosphate group here. All right, my sugars here, which in this case is deoxyribose because it's DNA. And then here is my nitrogen base. Okay, and what I want to point out here is take a look that the phosphate is bound to the sugar, is bound to the base. That makes a full nucleotide. That phosphate then attaches to the sugar of the previous base, and the sugar attaches to the next phosphate of the next nucleotide. And the reason that this makes a difference is because there's going to be directionality to the way that this strand works. If you take a look at this, one end of the strand ends with a free sugar, right, that's not attached to anything. The other end of the strand ends up with a free phosphate group. And so we're going to talk about these as 5' versus 3' ends to the molecule. So this is my 5' with the free phosphate, excuse me. And then your 3' end is down here with the free sugar. Now, why does that matter? Because we're going to look at the double strands of this molecule, and one strand is going to be going 5', follow that guy all the way down to 3'. But these molecules are going to run what we call anti-parallel to each other, and that's going to be really important when we get to talking about DNA replication. So while that strand runs 5 to 3, its opposite strand starts 3 to 5. And that's going to end up making some complications, if you will, for how this works. So just kind of keep that in mind. The numbering convention comes from, if I go back here to this, right? 5', it's your 5th carbon that phosphate is attached to, and 3' is down here where another phosphate could potentially attach if there was another nucleotide. That's sort of our bonding site. But if there's nothing attached to it, that's where that 3' comes from. Okay. A couple other things while we're here in terms of structure that you should be reminded of or just remember. So when we look at those complementary base pairs that are held together, right, they're held together here in the center by hydrogen bonds. Okay, so there's hydrogen bonds holding those bases together. And remember, with C and G, they make 3 hydrogen bonds. That's why they want to connect to each other, and A with T can only make 2. The other thing to keep in mind is when we look at the types of covalent bonds that are holding that sugar and phosphate backbone together, holding one nucleotide to the next together, we talk about those phosphodiester bonds. So when I'm looking at this here, right, this right here would be a phosphodiester bond. And the reason that's going to be important goes back to this whole one strand, one's run direction, one runs the other. We'll talk about the fact that the strands run anti-parallel to each other. So one going one direction, and then those phosphodiester bonds, basically the directionality is flipped when we get to the other strand.